Hello, welcome back to my other video. Today, we'll talk about the Calvin cycle and photorespiration. But before we discuss it, have you subscribed to my channel yet? If not, then please subscribe and click the notification bell icon for future videos. Alright, are you ready? Let's get into the discussion for Calvin cycle. The overall purpose of the Calvin cycle is to convert carbon dioxide from the atmosphere into carbohydrates or sugars, which the plant needs to power its cellular activities and build new plant structures. The Calvin cycle reaction occurs within the stroma of the chloroplast. The reactions use ATP and NADPH produced by the light reactions to convert CO2 to carbohydrate. Let's zoom into the Calvin cycle to take a closer look. The Calvin cycle can be divided into three phases: carbon dioxide fixation, carbon dioxide reduction, and regeneration of RuBP. The first phase, carbon dioxide fixation, basically captures the CO2 from the atmosphere, so it can be used in the reactions. To do this, CO2 is attached to RuBP, a 5-carbon molecule. The enzyme used in this reaction is called Rubisco, and the result of the reaction is an unstable 6-carbon molecule that quickly splits into two 3-carbon molecules called 3-phosphoglycerate or 3PG. Next, the process of converting the CO2 to carbohydrate begins. This phase is called the carbon dioxide reduction phase because we're adding electrons and energy to the CO2 molecule. During this phase, the sequence of reactions uses NADPH and some of the ATP from the light reactions. These molecules supply the needed electrons and energy for CO2 reduction. Electrons are added from NADPH, and through a series of reactions, 3PG is reduced to form G3P, a carbohydrate. ADP and NADP plus return to the thylakoids to be converted back to ATP and NADPH by the light reactions. One of the G3P molecules is set aside as a building block for glucose, but the majority of the G3P molecules move forward into the third phase of the Calvin cycle. In this phase, ATP is used to combine the rest of the G3P molecules to form RuBP molecules. This RuBP can then combine with additional carbon dioxide molecules, continuing the carbon reactions. To form a glucose molecule, the cycle actually has to turn 6 times, because each turn of the cycle adds only 1 carbon atom from the incoming carbon dioxide. Remember that Calvin cycle is dependent on the light reactions which provide NADPH and ATP. Now, let's talk about the photorespiration. Recall that the chloroplast has 3 membranes: the outer membrane, inner membrane, and thylakoid membrane. The compartments include the stroma and the thylakoid space. Plants in hot, dry environment lose a significant amount of water through their stomata. Essentially, plants become dehydrated to prevent water loss. These plants close their stomata. While the stomata remain closed, photosynthesis continues in these plants. Recall from the previous lecture, photosynthesis consumes carbon dioxide and produces oxygen. Normally, the oxygen produced by photosynthesis diffuses out through the open stomata. But when stomata are closed, oxygen builds up and carbon dioxide decreases in the plant leaf. With an elevated oxygen concentration, Rubisco in the stroma will attach oxygen rather than carbon dioxide onto tribulose biphosphate after the attachment of oxygen onto ribulose biphosphate. 2 molecules result 3-phosphoglycerate, which is a 3-carbon molecule, and phosphoglycolate, a 2-carbon molecule. Phosphoglycolate cannot continue in the Calvin cycle. Thus, it is broken down and its carbon units are released as carbon dioxide and wasted due to insufficient carbon in the Calvin cycle. Glyceraldehyde 3-phosphate or G3P cannot be produced. However, the cycle does continue and ribulose biphosphate is regenerated. This process is known as photorespiration. In summary, photorespiration wastes carbon, requires energy input, and does not result in the formation of glyceraldehyde 3-phosphate, a precursor of glucose. Thank you.